But how do we control this? This is just silly. I have a feeling it's something really simple.
I wonder where's just this. So
you got it to go. The thing is actually on.
Now there's a remote there that's the Panasonic.
Oh, yeah.
Gum on it is the side. But the Panasonics are that. The Panasonics.
So it's warm enough, so I should be able to see it.
Yeah.
Center, but it's on center. But
And how is it off?
this is on. Now that went off. Because it should show up now that that's showing up. But how do we turn the side ones on?
The side projectors are on. You can see the lights are on.
Right. But okay, side laptop. This is set the laptop to display. Okay. There are no controls. So why doesn't it?
This is this one.
Well, see, we don't usually use it. That's the thing to do. So I don't think we normally use it.
See the this is the first side.
But I can't use it. You can do it. I mean, it's on.
Just
Maybe give it a chop top.
this is just bullshit.
Yeah, no. Well, you can't find him. So, we ought to get the other guy, Charlie. But this is just crazy. This thing is not working.
You've got two projectors.
Right.
So you can operate.
Side laptop. And then you get this.
Why is it?
That's what I don't get. See that? Reboot. I mean, there's the console.
Yeah. Look.
This is just.
What? Why?
Oh God, it's 5 minutes later. I guess I can. You can go ahead and do your, because you're not. Yeah, there's nothing else for you to do. Okay, can I have your attention? We're having equipment problems, but I wanted to give Dr. Boyd Butler a chance to talk about a new course that we're offering.
So in the spring, we're going to offer a class called Experimental Cell Biology. This class hasn't been taught for quite a while, and so it needs to get back on the books. Basically what this will be is, in many ways, a continuation of this cell biology class. Many of the things that you go over in class, we're going to discuss the techniques that people use and do use to study these things, transcriptional activation, signal transduction, and these sorts of things. Everything from molecular biology techniques, cloning, subcloning, RNA knockdowns, RNAi, just standard techniques, westerns, northerns, southerns, some of the new microscopy techniques, fluorescent microscopy, FRAP, confocal, things like that. So it will be both a classroom-based. The classroom-based will be the minimal aspect of it, but it will be a lab-based course. And what we're planning on doing is, for the first third to half of the course, we're going to teach you how to do these techniques. Teach you to basically have an in-class demonstration. Everybody will be able to do the techniques themselves. And the last half of the class, what we're planning on doing is having individualized projects, where basically you're kind of on your own. You have a project to take this particular gene and subclone it and do all everything that you've already learned. Now, obviously, if anybody spent any time in the lab, things fail all the time. So that won't be your grade won't be predicated on whether things work or not all the time. But I think it'll be a good hands-on experience. It's called 3310 Experimental Cell Biology. If you're interested in taking it, it'll be offered in the spring. Go see Pam, go see Sam, and we're looking for, I don't know how many we're looking for to make the class. It would be good for people. I don't know if you're planning on going to grad school, even if you're planning on going to med school. These days, having been there, these days research is extremely important in all aspects, both in medical school and obviously in graduate school. One of the things is if you have these research experiences, if you go to grad school or you go to medical school, and you want to do research, you have a big head start on people that don't.
Yes, sir.
So we'll teach you all the basic techniques. So if you're interested, go see Pam and Sam, and maybe we'll see you in the spring. So thanks.
Thank you. And next fall, you can take Cell 2, the lecture course.
Yes.
It's team talk. Oh, here we go. Now we have a whole team of people working on the patient over here. Okay, so a couple of announcements while they're getting this up to snuff. The first one is, by Friday, since I gave you an extra week for the test, what I want you to do with your group is by Friday to pick a broad topic that you're going to work on in terms of your poster project. Okay, so I've explained that on Piazza, and so go to Piazza. The details are there. I don't want to repeat myself. But it's a broad topic: cancer, diabetes, Alzheimer's. But what you can't do is single gene problems. I don't want projects recycled from your genetics class. So that's the biggest restriction. It needs to be a polygenic origin type of disease that you're going to attack. Okay. So that's by Friday. And I know you'll be meeting with your groups this week for study or whatever for the test, which is in 1 week from today. We stopped the material with Lecture 6, and so Lecture 7 is completely new material, not on Quiz 1. And so we're going to finish up a little bit of, we got into Lecture 7, but we didn't finish it. So we're going to finish Lecture 7, and then we're going to get into membranes, okay, which is the next topic in the course. The other announcement is for the graduate students. Remember, your papers are due, your choice of 3 papers are due to me by Wednesday. And where's Daniel? Daniel? I need yours really quickly because you're one of the first ones. So get me that choice, and I've already picked Nancy's, and so we can get that PDFs out to the graduate students. We won't have a meeting again this week or next week, because we never meet on the weeks of tests, but the following week, so 2 weeks from Wednesday, is our first presentation. And it looks like the patient needs CPR and is not going to get it. Okay, so we were talking about various ways, so it's going to have to be a chop talk until we get something going here. We're talking about various ways to control gene expression. So we've actually moved out into the cytoplasm, and we looked the most recent technique that we looked at for controlling gene expression is actually moving the messenger RNA around. Okay, that happens to be one use of the 3' untranslated region. There are signals, they're actually controlled RNA elements that allow it to be grabbed and moved to a particular location. Okay, so what other kinds of regulation of gene expression can we use? Well, let's talk about translational control. So that's a big piece. That is, you control which proteins are synthesized and how much. So there's two types of controls for translation that occur in the cytoplasm. One is global. That is, we're able to globally speed up or shut down translation. So the speed of all protein synthesis goes up or down. And then there is specific translational control that's invested in particular proteins. And I'll give you an example of each one of those. Okay. So let's talk about the global control of translation. Let's take a red blood cell that has one job and one main job, and that's to make hemoglobin, right? In order to make hemoglobin, what you need is a prosthetic group that actually carries the oxygen, that carries the oxygen. But the heme group is not a protein; it's a non-protein piece. If that heme group is missing, the hemoglobin can't carry oxygen, and so we need to suppress protein translation, because why make a bunch of hemoglobin if you don't have the heme group to go with it to carry the oxygen? Okay, so that makes sense. This is one of those rare cases where you want to suppress all translation. Normally, suppressing all translation would do what to a cell? You kill it, right? So normally, this is not something you want to do. But if you want to do that, what you do is you work at the level of the translation initiation complex. That complex is composed of three things. And I'll turn this on. So to initiate translation, as you know, you need eukaryotic initiation factor 2. Right? And I won't say the E anymore. Let's assume that we're all dealing with eukaryotic cells, as we have been throughout this course. So initiation factor 2, you need some GTP, and you need the initiator transfer RNA. Okay? That happens to be the same one that carries methionine. So the first amino acid is always methionine, with a few exceptions. So these three things need to come together to bind to the small ribosomal subunit, and then it hunts for the large ribosomal subunit, and then you initiate translation. So if you can inhibit this part, then you can stop translation. And that's what we do. If you can phosphorylate this, put a phosphate group onto this, you can actually turn it off. Okay. And there is a kinase that does this, and it's called HCI. That's not HCl, that's HCI. It's heme control inhibitor. And that's in your notes, so you don't need to copy that down. Heme control inhibitor is itself controlled by what? Well, if there's lots of hemoglobin, the hemoglobin, let's just call it heme. Well, the heme actually is it's not hemoglobin, it's the heme prosthetic group. Will bind to HCl and turn it off. It goes off. So lots of heme group, we don't want to suppress translation. We want to keep it on, so we want to turn the kinase off. Okay? But what if you start to have a depletion, you don't make enough heme group? Let's say you've got iron poor blood, you don't have enough iron. There's lots of problems that can cause that. And so if there's no heme group, what happens is this is freed from the inhibition, and it turns on. Okay? And so it's a kinase, and so kinases do what kinases do, that is to put a phosphate on the protein. What protein does it phosphorylate? It phosphorylates initiation factor number 2. Okay? And that turns it on. Okay? Now, that's kind of where your book leaves it, but we need to go a little bit further into this and understand, how does phosphorylation turn off IF2? Because it's our very first example of a GTPase. This eukaryotic IF2 is a GTPase. And that means it is itself a molecular switch. The same way that phosphorylation and dephosphorylation can turn proteins on and off, so can picking up a GTP and losing a GDP. Okay, so here's the cycle. To turn, so this is IF2, okay, and it's off when it's bound to GDP, Okay, it's in the off state. And so it's just a guanine nucleotide binding protein is really what it is. All right? To get it to turn on, what you have to do is you need to bump into a protein that's called a GAF. The GAF stands for guanine nucleotide exchange factor, because we're going to make it lose the GDP, okay, and pick up a molecule of GTP. Yes, they fixed it. Okay. So what happens is when it picks up the GTP, IF2 bound to GTP is on. Okay, it's on. And then it's going. What is it going to do? It's going to bind to the initiation transfer RNA, small ribosomal subunit, do its thing, right? So if you want translation, you need to have this thing turned on. If you want it to be suppressed, you want to turn this off. Okay? And what happens, just to complete the cycle, how do you get back to the state where it's turned off? Well, what happens is it bumps into another protein called GAP, and it's not the gene store. It is wanting, it's GTPase activating protein. GTP. So what it's doing, this protein actually is an enzyme, and it's quite capable of splitting GTP. And when it does that, it releases a phosphate, and it returns to the GDP state. So you need to learn this cycle now, because we're going to use it over and over and over and over. So the specifics are a little bit more complicated, and what I want to do is turn on my pictures, so I'll show it to you more completely. So let's take a little break while I do that, all right? Yeah, but you can ask me questions. Everybody okay with this switch mechanism so far? Now, yes?
So, I'm confused. You said that the HCI inhibits the translation issue, right?
Right. It inhibits one thing. It inhibits IF2. And that's what we're talking about. IF2 is a molecular switch. And so what you're doing is somehow it's turning it off, and you can't follow that rest of the story until I get my picture.
You turn it off when there's not heme present.
Exactly. Because you don't want to make a bunch of hemoglobin and then have no heme combined with it, because that's not energetically favorable. You'll waste all that energy. Because eventually the hemoglobin will be degraded, and you synthesize hemoglobin for no reason. Right? All right, so let's take a minute. I turn this off. Talks amongst yourselves. It looks like we only have one. Okay, so that's the topic that we're talking about. Yeah, for some reason, this guy is not showing up. Maybe I need to just take it out and put it back in. Boy, you're going to have to show me what they did because I've never seen that. That helps. Oh, okay. Good. I'm okay with that. Thanks, Charlotte. Okay, so we're back. All right. So this is what we were talking about, controlling this guy right here. And so let's say that we're not making heme. So this HCI, heme-controlled inhibitor, turns on, phosphorylates it, and somehow it's going to turn it off. So we have to understand that. I just showed you how this turns on, and that's through a GTPase switch. So what happens is it has to bump into something called a GEF that will make it lose GDP. It loses the GDP, and then it picks up GTP, and it transitions into the on state, right? Well, this protein that makes it do that is called a GEF, and it's actually eukaryotic initiation factor 2B. So it itself is a protein. Now, what happens is if you phosphorylate this guy, and this is just the switch, shows you all the components. I just drew that for you. Okay, so that's drawn out. So what happens is if HCI phosphorylates IF2, what happens is you put a phosphate group on there, and then when IF2B, which is the GEF, bumps into it, it'll actually grab onto it and won't let go. So it actually sequesters all of the IF2B in a bound state, and IF2B cannot go around and turn on other initiation factor proteins, right? So it's all stuck together, and that is what suppresses protein translation. Because you've sequestered all of this stuff, without the GEF, you can't turn on IF2, and so it shuts down. So the mechanism is to actually sequester all of the GEF, guanine nucleotide exchange factor, and prevent initiation complex from ever forming. So this shuts down globally in translation. It turns it way down. Okay? All right, so any questions about this so far? Yes?
The gap. Gap, you don't know gap.
The gap?
Gap when it's not.
That's the gap. Okay? And I'm not going to say what protein it is, but it's a separate protein. The critical thing about this type of GTPase, we call them small GTPases because it does not carry its own gap. There are other proteins that the GTPase switch has a bigger piece to it that actually carries its own gap with it. Okay, but we'll talk about this later. All right, so that's the mechanism. Now let's talk about a specific mechanism of shutting down translation, not of all of it, but of a particular a particular protein. And in order to get that story and tell it well, we need to understand a little bit about iron metabolism. So we're going to talk about inhibiting or stimulating the translation of two particular proteins that are involved in iron metabolism. Okay, and that's what your book uses. So if you want to absorb iron, what do you use iron for anyway? Why do you want iron? Carry oxygen, so it's a component of hemoglobin, right? But anything else? Even if you're not a red blood cell, why do you need iron? I'm sorry?
What if you use this part of the catalyst
Think about where you use it to move electrons, okay, to carry electrons. And where does that happen all the time?
And the electron transfer chain in the mitochondria.
In the mitochondrial electron transport chain. So iron, sulfur clusters are used to move electrons down that chain, and that's what produces a proton gradient that gives you ATP. Okay, so iron is really important. So the cell is like all over it. If you're not making enough iron, it's going to do something to make iron be retrieved from the blood. So iron is absorbed, let's say, the 3+ oxidation state. This is absorbed by your gut, okay? And so then it gets into the bloodstream. And you never let iron go by itself. Iron's very toxic, and so generally it's carried by a protein to protect it from the creating toxicity problems. Okay, so in the blood, it's actually carried by a protein called transferrin. The ferrin word tells you it's carrying iron, which is ferrous, and transferrin means it's a transport protein. So transferrin is a protein that is bound to iron, okay? But transferrin is too big to go into a cell by diffusion. So it gets in there by receptor-mediated endocytosis. So there must be a receptor for transferrin. And we call it, strangely enough, the transferrin receptor, all right? And it's a single-pass membrane protein, which we're going to talk about today. So this is the transferrin receptor. And we generally abbreviate the word receptor by RC, receptor. Okay, so the transferrin goes here, hitches a ride, it's carrying Fe. This gets internalized, okay, by endocytosis. It goes to the lysosome, and that produces a membrane-bound vesicle, and it ejects the iron into the cytoplasm, and then it binds the hemoglobin, or it goes into the mitochondria and does its job there. Okay? So, two proteins you would want to upregulate if you're starved for iron is, number one, transferrin. But the one we're really going to look at is the transferrin receptor. You can't get transferrin to come in unless you have a receptor. And it's dragging its buddy iron along with it. Okay, so those two we want to upregulate. Now, which protein would we want to suppress if we've got too much iron? Let's say you've eaten lots of protein. You went to the Hub City Barbecue Festival last Thursday and ate lots of ribs and things. It was pretty good, of course. And so you have lots of iron, but iron is toxic. Okay, and if you get too much iron in the cell, what can you do with it? Well, there's a protein called ferritin. There's that word again. Ferri means that it's a iron binding something. Okay, ferritin is a cytoplasmic protein that specifically has a binding site for iron. Okay, so it's got a little binding pocket, and so it's just a sponge. It soaks up all the iron. And then once the iron levels start to fall, ferritin can release it at small doses to give it back. So it's a storage form for iron. So you want to upregulate ferritin when you have too much iron, and you want to suppress ferritin synthesis if you don't have enough. Because the last thing we want is ferritin around soaking it up if we're trying to pull it in from the blood, right? So there's two different physiological states: iron starvation and iron excess that we're going to actually look at. Okay, do we all have the physiology correct? Because that's where you start when you're thinking about this problem, is what am I trying to suppress or induce, and why am I doing that? Okay. All right, so we have that down. Now, let's look at the mechanism. Okay, so this is kind of cool. So let's talk about the situation where iron, we have low iron. Let's say you're a vegetarian and, or whatever. Vegan, you're from the planet Vega, and your iron levels are really low, okay? So what happens is this protein, which is called iconitase, it's a cytoplasmic protein, will actually bind to the messenger RNA that's going to make ferritin. And where does it bind? It binds to a control element. So again, here's this concept where RNA contains control elements. It's not just this piece of stuff that's read by the ribosome. It's got control elements in both flanking regions that are on either side of the protein coding region. Okay, so the 5' untranslated region is actually a control sequence. and it can actually base pair to itself. This is called a stem loop structure that you're very familiar with when you study transfer RNA, because transfer RNA has 4 different stem loop structures. So this base pairs to itself and forms what's called an iron response element. That word makes sense, right? Because it's a nucleotide sequence that's a control element. So it's called the iron response element. And that's the thing that aconitase recognizes. So under low conditions of low iron, aconitase will bind to this and form a block to the initiation of translation. Not of all messenger RNA, just this one. Okay. The reason this forms a block is what has to bind up here in the 5' region? It's actually, it recognizes the 5' cap as a signpost for where it's supposed to bind. What part of the ribosome binds first? Small subunit, right? And so the small subunit binds and it scans along here till it finds the start codon, and then it has to pick up the transfer RNA met. It's got to pick up IF2 initiation factor. So it can never make it to the start codon because of this big bulky thing. So that blocks the scanning of the ribosomal assembly. Right? So if you block that, you will shut down the translation of the ferritin messenger RNA only. Okay. Now, what happens if you take an iron supplement, you take a vitamin, you can't eat protein if you're vegetarian, and your iron levels go up in the blood, and it gets pulled in for reasons that we'll talk about. You have the transferrin, binds to the transferrin receptor, brings it in, and so iron levels start to go up in this situation. What happens is the iron binds to aconitase. The aconitase has an iron binding domain, and when it binds to the iron, it changes its conformation. So aconitase dissociates from the response element because it's changed its shape. Right? And if it lifts off and is now sequestering iron, which is what its original, this is one of the regulators of aconitase, then that frees up this 5' untranslated region to have a ribosomal small subunit bind to it and scan up and down and find the start site. So you get rid of the protein, and then this thing is able to scan through this stem loop structure. Okay. So again, the big concept here is how do you specifically inhibit one messenger RNA and not everybody else? You target the 5' untranslated region. Okay, and it's controlled by the physiology of iron concentration. Okay? Good. Now, if there's control units here, there obviously is going to be control units in the 3' untranslated region. And we already know that. There's a control element, in the 3' untranslated unit region, 3' untranslated UTR, that binds to a mitochondrial a motor protein that we talked about last time. So we already have that under our belt. But is there anything that affects the stability of messenger RNA that's located in that 3' untranslated region? Well, the answer is yes. Okay. The 3' untranslated region is attached to the poly A tail, as you know, and the poly A tail is never naked. It's never exposed sequence of adenosines. It's actually covered with a protein called poly A binding protein, PABP. It's actually PABP1. Okay. And so every 30 amino acids, 30 nucleotides is covered by a little piece of protein. Okay. And that goes into the cytosol, and that tail is continually being shortened with time. So it's like a burning fuse. And what happens is, as this gets short, and you reach less than 3 of these adenines, the poly A binding protein is now lifted off, and that exposes the 3' UTR. And what happens is, first of all, the 3' end of this is actually coupled to the 5'. It's actually a cyclic molecule. The 3' end is touching the 5' cap. If you remember from the video, that's the first thing that happened to the messenger RNA after it went through the nuclear pore, is it turns into a circle. Okay, and I'll explain why in just a moment. But the first thing happens here when this goes too short is this thing gets decapped. There's a decapping enzyme. And if you decap the 5' end, then that's just like open season on messenger RNA. It gets chewed by an exonuclease. And not only from this end, once you start chewing at the 5' end, it starts to chew from the 3' end. So it's in fact chewing at both ends, and this thing is quickly degraded. So a couple of important things here. The 5' cap is important in order to keep the messenger RNA from being destroyed, and so is the 3' end, because it's what is attached to the poly A tail. The poly A tail is a burning fuse that when it gets too short, you're going to lose the messenger RNA. So the control of RNA stability is sort of in this little area right here. Okay? It's in that little area. And what happens, and we're going to talk about a couple of things that, well, here's the story about why it's important for the 5' cap to be touching the 3' poly A tail. When they're touching each other, what they do is they actually recruit the initiation factors that are required for translation. This is bound to poly A binding proteins, and the poly A binding proteins in the 5' cap recruit proteins that say, hey, I want to be translated. And so all the initiation factors come to bind to the 5' area, and then you zip through this over and over and over with what? A ribosome. And that's kind of cool because the ribosome comes through here, right? It hits the termination codon, and what happens to it? It what?
It pops. It's closer to start to code on than it.
Well, the two subunits do pull apart, but since they're right next to this, they can reassemble very quickly. So there's a pool of ribosomal subunits always being reappearing at the 5' end, and so it can start over and do this over and over. So here's the thing. Translation prevents RNA degradation. Why? Because these ends are always protected. So there are two competing processes here. RNA translation, messenger RNA translation, and RNA degradation. And if the degradation wins, what happens is this thing gets decapped, as I told you. This is too short, and so then the 5' cap actually loses the proteins that recruit those initiation factors, and instead they recruit a D-adenylase. That's the enzyme that chops off all the A's. So when the rate of synthesis slows, you're going to actually cause the rate of degradation to go up. They are coupled. Questions about the coupling? Okay. But one of the control elements that control stability are usually found in the 5', the 3' and translated region, not the 5'. Okay. So let me give you a specific example of a control element. And we're going to talk about a different protein, but you already know this protein because we talked about the iron physiology. So this is the messenger RNA, not for ferritin, which is the other one we were looking at, but for the transferrin receptor. Okay. Now remember from the physiology, if you're in iron starvation state, that means you want to suck all the iron out of the blood that you can. So you want to stimulate the translation of the transferrin receptor. That makes sense. So get that pattern in your head correct first. Under these conditions, what do I want to do to this messenger RNA? We want to stabilize the messenger RNA, right? Because we want to make lots of it. So to do that, we have another iron response element located in the 3' untranslated region, and it recruits the same protein, a conjugate. And what happens is this thing prevents this from being chewed up. It's like a big block to that D-adenylase, right? So this is actually going to stabilize the messenger RNA, and if you stabilize it, it just means the half-life lasts longer than it would otherwise. So as long as that's hanging around, you can be making more and more transferrin receptor. Okay. And it's clear why this would create a block. You can't get in here and chew off the 3' end. Right? It's too sterically inhibited. Now, what happens when we raise iron? Take a guess. Iron is going to bind right there, right? What's going to happen? It's going to do the same thing it did on the other messenger RNA. It's going to lift off. But in this case, what happens is when it lifts off, it doesn't stimulate translation. It inhibits it because you're going to chop into, what happens is when this thing lifts off the iron response element, there's a sequence of AU in here, nucleotides, that makes it really juicy to be sliced apart. It makes it really susceptible to cleavage. And so those AU sequences are like, come chew me up. So when you expose them by dissociating a conatase, this thing gets rapidly degraded, it gets decapped, and then it gets chewed from both ends very quickly. Yes?
Is it the same iron binding site as with the ferritin?
Yes. Well, you mean on the conatase? Yes, it's the same protein, the same binding site. And so, but don't get confused. In one case, the lifting off of a conatase stimulates translation if it's at the 5' end, and if you lift off here, you're going to stimulate degradation. You're still stimulating something, but it's a different thing. Right? So if you put all this together in a big concept, is there's control elements located at 5' and 3' ends. So those are not junk. Okay, they're control elements that control the rate of translation. Okay. That makes a lot of sense. All right. So we talked about mRNA stability and all these, the details are here. So then we come to a major topic, which is really interesting, and that's the microRNA information. So microRNAs is another way of controlling translation. And actually, it goes well beyond this. microRNAs were discovered in 1998, so not too long ago, by Andrew Fire and Craig Mello. 1998, 8 years later, they had their Nobel Prize. That's how important this is. So usually it takes years and years and years for people to figure out that you're a genius. It took them 8 years to get the Nobel Prize. So it's really critically important. So where are we along this control path? We're in the cytoplasm now. We're talking about things that control translation, and actually specifically messenger RNA degradation. So what are microRNAs? There's about 1,000 microRNA genes scattered through the genome that we know of so far. There might be more of them. They look like a standard protein coding gene. They have promoters, they have a, what looks like a 5' translated region. They're fairly short, but they're actually translated, they're actually transcribed by an old friend of mine, Pol 2. So it gets capped when it comes off the gene. So a microRNA gets capped and it gets polyadenylated, because like any self-respecting RNA, it will die very quickly if you don't coat it with protein. So the poly A tail is coated with the poly A binding protein. The 5' end is capped, and maybe you didn't know this or remember it, but all along the RNA there are SR proteins that block access to the RNA so it cannot be cleaved. So it's heavily coated. It zips through the through the nuclear pore, gets into the cytoplasm, which is the site of translation. Okay, so that's where microRNAs are going to do their job. And this is, let me move this right there. Okay. So this is a little bit complicated, and it gets more complicated every year that we figure out more stuff. So your book came out in 2008, and the new editions actually come out next year. And so I've had to update this, just a really basic. There's a lot of other stuff that we know about already, but here's the basic idea. So you take a microRNA gene, you transcribe it by Pol 2, okay? And immediately what happens is it forms a base pairing hairpin structure. This is a stem loop. So it's the RNA base pairs to itself in specific regions, like this. And so there's a part where it doesn't base pair, but all along here it forms this loop, this stem structure, and then there's another open thing. That's not important. But this is called a hairpin loop. Okay? And there's the cap, and there's the poly A tail. This all occurs in the nucleus. Okay? And then it's going to get cleaved. It's going to get cleaved. And so this thing that comes off the gene is called the primary microRNA. And so that's that word pri. It's not pre, it's pri microRNA is this thing that forms the hairpin loop. And then there's a protein that's in the nucleus. Well, remember, it is a poster child. It is in the nucleus. It is a nuclease. And it's going to cleave this primary microRNA right there, cleave off that tail. Okay? So then that little piece comes through the nuclear pore, and we've got double-stranded RNA. That's the key. Double-stranded RNA is a signal to the cell to do something. Not make a protein. It's going to regulate actually transcription and translation. Okay, so we have this primary messenger RNA that pops into the cytoplasm, and that's where it meets up with another protein called dicer. So this is sort of slicer, and this is dicer. Okay, dicer is another nuclease, and what it's going to do is cleave off this little loop right there. It's going to cleave it off. All right, and so what is actually left is a double-stranded RNA that's 21 to 22 nucleotides long, base pairs long. It actually has a 3' overhang at both ends, so it's got like a couple of nucleotides that are not bound to anything, that are not hybridized. So it's a short piece of double-stranded RNA. Now, what creates that is dicer. So you slice this and you create pre-microRNA. That's the 22 nucleotide sequence. So that's the pre-microRNA. That is then loaded onto a complex of several proteins that we irritatingly call RNA-induced silencing complex, or the RISC complex. RNA-induced silencing complex. The thing that is the business end of that RISC complex is a protein called arginine. Okay. Now, how do you get that pre-microRNA loaded onto the RISC complex? Well, there's two proteins that help. Dicer not only cleaves and trims it so it becomes a pre-microRNA, it actually is a helper that's called R2D2 because biologists have a sense of humor. All right? So these two guys loaded onto arginine. And so arginine now is sitting there with the double-stranded microRNA in it. Okay? And then arginine recruits another protein that I think actually, in your PowerPoint, I think this label is 3CPO. I mean, Darth Vader would have my head for that. But it's actually C3, it's actually component 3 promoter of RISC. That's what it is today. Okay, so C3PO has a very important job. Before you can build a microRNA, which is a single-stranded piece of RNA, you've got to chop off the other strand. Who does that? That's what C3PO does. It nicks the passenger strand. Okay, so there are two strands in microRNA. There's a guide strand, that's the business end, and then it's base paired to 22 nucleotides called the passenger strand. That's the one we're going to chew up. So that's what C3PO's job is, to nick the passenger strand and cause that single strand to be degraded, leaving an intact guide strand. So when RISC is completely activated, what's it got? It's got a single strand of RNA, microRNA, that's called the guide strand, and then this RISC complex can go do its job. So are we following this along? So we started in the nucleus, we transcribed, transcribed the RNA. It came off the gene. That transcript is called primary microRNA. It gets chopped by Drosha. Okay, that cuts it down to the pre-microRNA stage. That's still too big, so we need to get rid of this loop. And Dicer does that, chops off the loop, and now it's a double-stranded microRNA. Dicer and RTD2 hand it over to arginine, so they load it on arginine, and then C3PO nicks the passenger strand, and now we've got a machine. We've got a protein complex, this little green guy. There's several proteins. We're not going to worry about all of them. But it's got a trailing guide strand that's going to be hunting for red October. What's it looking for? It's looking for messenger RNA. Okay. And there's actually another protein, and it's called Luke. It's luciferase, don't write it down, luciferase urea stable kinetic enzyme. We don't know what it does, but we know that Darth Vader is its name. So I'll update that story at some other point. Okay, so this is in the cytoplasm, and so this little thing, it's got a piece of RNA. It's hunting for a sequence that it can hybridize to. What is it hunting for? It's hunting for a messenger RNA that has that sequence in its 3' untranslated region. So again, this makes perfect sense. This thing is going to look for a sequence that it can latch onto in the 3' untranslated region. Okay? So there are two possibilities. Now, if this guide strand perfectly hybridizes to the 3', it's a real short sequence. The guide strand is a short sequence. And so if you perfectly hybridize to an exact complementary sequence, what happens is Argonaut right there and there chops the RNA and kills it. Okay? If you cut it, what's going to happen? Exonucleases are going to chew it to pieces. Okay? So Argonaut will do that. Now that's what happens typically in plants. Okay? Plants, the microRNAs, and it was actually discovered in plants or roundworms, I can't remember. But the plant, that's what happens in plants. It just, Argonaut goes at it right there. Chops the target messenger RNA, translation is completely inhibited. Now, in us, what happens is typically that guide strand does not form a perfect match. You can see that this right here, this little loop, is a place where the RNA does not hybridize to the messenger RNA. Okay? But that's okay, because what happens is it inhibits translation instead of cutting the messenger RNA. It stops translation. Okay. What does it do to it? Well, it stops translation, and then it gets drugged, kicking and screaming, to something called P-body. P-body stands for processing bodies. And there are little places in the cytoplasm, very organized, where all the decapping enzymes and the deadenylase live. Okay? Processing bodies are just localized regions where all those enzymes like to get together and have coffee. Okay? And so the messenger, the microRNA, the RISC complex drags it to the P-body, and then what's going to happen to it? Okay. So let me turn the lights down a little bit. So if we attach a fluorescent fluorochrome to the capping enzyme, it shows up in the cytoplasm with this distribution. It's localized in these little dots. These are the P-bodies. Okay? Now, if we put an antibody and we label it with rhodamine, which is a red fluorochrome, and attach it to an antibody that recognizes Argonaut, we get this distribution. Does it look very similar? Absolutely. That's because Argonaut is a component of RISC, and the first thing that RISC does to that trap, that ensnared messenger RNA, is to drag it to the P-body. And if you superimpose those two, and they're exactly the same place, you should go from green to red, you should get a yellow color, because that's in between red and green. Okay? So that shows that the P-body is where everybody goes to have fun and chop up the messenger RNA. Okay? Questions about that? So this is really cool. What's interesting about it is you can have a single microRNA that can control entire families of transcripts, because it doesn't have to recognize the entire sequence of the messenger RNA. All it needs to do is find about a 7 base pair sequence in the 3' untranslated region, and it can control a whole group of transcripts. So this is really important in development. Remember the homeodomain proteins that cause your body plan to be what it is? Okay? That transcription factor is going to turn on 10 or 100 proteins. They're going to have all these proteins that make where your arm comes out. It's not one protein. What if you wanted to shut that down so that the arm doesn't grow out of here? Okay, you want to shut all of those messenger RNAs down in the wrong place. You can use a single microRNA. It's really very efficient. Now, the way you don't even need just one. Sometimes the mismatch is so great that it's not enough to stop translation. But guess what? You can have a pylon. Okay? You can have more than one microRNA attacking the 3' untranslated. So these guys get combinatorial. That is combinatorial control over messenger RNA degradation. So that combinatorial thing is a central concept in gene control, whether we're talking about transcription or messenger RNA translation. Okay, so any questions about that story? So this is the microRNA story. And this, what happens is this turns out to be of great therapeutic utility, where eventually we'll be able to take microRNAs, target them to, say, cancer cells, and turn off a particular enzyme that's causing that cell to replicate endlessly. Okay, so that's the new therapeutic target. And it's not just cancer. We can do this with many other types of things. So it's what happens when you turn down the translation by chewing it up with a microRNA, that's called a knockdown. It's not a knockout. A knockout is when you destroy the gene. A knockdown is when you dampen the translation by using a microRNA or any kind of double-stranded RNA. So if you hear knockdown experiment, that's what they're talking about. Okay? Now, there's one other, oh boy, well, we started out late, so. Now, you can actually, there are other types of genes that produce what's called small interfering RNA, and as investigators, we can synthesize these in the laboratory. So if I want to inhibit that particular messenger RNA, I can look at the sequence, the 3' untranslated region. I can synthesize one of these, turn it into double-stranded RNA, and inject it into a cell, and by golly, this is what's going to happen. Okay? So however you get it, whether it comes off of a gene or we insert it into the cell ourselves, or a virus injects it. This probably evolved as a defense against RNA viruses. Okay? That makes a lot of sense. So this gets in the cell. This is like warning, to the cell. If there's double-stranded RNA, something needs to be turned off. Right? Now, you have two choices with small interfering messenger RNAs. Again, they are recruited to the wrist, and that's where they're chopped and trimmed. Okay. And then what happens is, if they can do the same thing as a microRNA, so you can have partial hybridization, block of translation, dragging into the P-body, so it does exactly the same thing. We don't need to list anything new. Okay. If it has perfect hybridization, what happens? Argonaut's chip, slices it right there on the spot. We don't need a P-body. But there's a third possibility, and that is the Ritz. It's not a cracker. Okay. This is RNA-induced transcriptional silencing. This is really bizarre. So what happens is this gets formed, and then this thing binds to a new set of proteins that cause it to form what's called the Ritz complex, and then it goes back into the nuclear core and returns to the nucleus. So now it's got this guide strand right there. And so it starts to look for the 5' translated region of newly synthesized transcripts. And if it makes a match with the 5' untranslated region of newly produced transcript, it will base pair to it and block transcription of that gene. What it does actually is it actually will recruit the nucleosome code writers to write it a code by methylating it that shuts the whole gene off and turns it into heterochromatin. This is bizarre. I mean, this is what happened. So you can actually use this as a way to stop a single gene, but also to heterochromatize a whole region of that chromosome. And this happens in the centromere. Remember, let's go back and think about this. And I'm going to need some help from Sabella, because it was her paper, and she took this class several years ago, that made this clear. Actually, that paper was particularly about meiosis, but there was a bizarre thing. Meiosis 1, meiosis 2 are not the same thing, and they're both different from actual mitosis. what they noticed was, I can't remember, I think it's mitosis 2, is it? That pole 2 becomes active. It associates with the centromere. What is pole 2 doing there? Pole 2 is there to transcribe, right? Why is it trying to transcribe a centrosomal region of the gene? Doesn't make any sense, right? It should be heterochromatized. Well, before it gets heterochromatized, what happens is there's a sequence in the centromere that pole 2 reads. It transcribes one strand, like the antisense strand, and it reads and transcribes the opposite strand. What does that give you? It gives you two pieces of RNA that are complementary, right? If you take a small sequence of DNA and you transcribe the bottom strand, and then you go and you transcribe the opposite strand in the opposite direction, those two strands of RNA, those two transcripts will then base pair and form a Ritz complex. That's what it does. So it actually is making its own double-stranded RNA, which in turn recruits the Ritz complex to come heterochromatize the centromere. It's really cool. So pole 2 is not always about turning things on. It can be used to make this thing happen, which is heterochromatization of a particular gene area. Okay, so that's kind of complicated. Basically, microRNAs and small interfering RNAs work by similar mechanisms. They just get produced differently. This is where this gets a little different. With a single, so small interfering RNAs can be used to activate a Ritz complex that actually shuts down transcription and causes heterochromatization. Okay, that was a lot. Questions about this? Yes?
Are you saying that all siRNAs are exogenous or they're endogenous siRNAs too?
I'm sorry.
Are all of the siRNAs exogenous or are there some that are endogenous?
There are some endogenous. There are siRNA genes. There are so
They
many.
follow that same pathway, the Ritz, the endogenous?
Yes. Yeah. And well, actually, I don't want to say that because I don't know that it does the hairpin loop thing. I'll have to check on that. Zisha and Shay, who teach the cell too, and actually in the spring, and teaches a whole class on this thing right here. I'll ask him to see what he says. Okay, we don't want to get that detailed. Okay, real cool. I think we'll just, unfortunately, let's skip the break because we had a big break at the very beginning. So the details are down here, obviously, in the outline. And so that gets us out of, we still have about 17 minutes, so don't get excited. So that gets us into lecture 8. And I want to, so that we're going to go into membranes now. This is a topic that you should know quite a bit about already. If you don't, again, I put together a PowerPoint and a outline. Come on. Oh, here we go. This is, yeah. That will bring you back up to snuff. I mean, you should know the fluid mosaic model backwards and forward. Who's heard of that? Ugh, it's so boring. Okay, but it's really not. You really need to understand why that model is important. Why is it important for the membrane to be fluid? Okay, so if you don't know, then I would say go back and look at that chapter. I think it's chapter 10. So we're not going to slog through every phospholipid structure in the book. Okay, you're supposed to know basically how they're constructed. And what we will focus on, though, is this guy here. I don't know what happened. Oh, okay, this didn't. Which one do I want to What? So, membrane structure, and so you need to review the basic properties on your own, but I will mention a couple of things to point out what's really, really important. And then we'll get started, but we won't get very far, with classifying membrane proteins into appropriate categories. So those are the two things that we're going to try to accomplish. This is the Chapter 10 membrane structure that we're not going to go through. You're supposed to know it. If you don't, just refresh your memory, okay? But it's essential that you know that background material. Like, this is the fluid mosaic model, okay? Why is it important that the membrane be fluid? Why? What are these little green things? Aliens? The proteins, right? These are phospholipids. It's a lipid bilayer, right? Why does this need to be fluid? You actually die if this is not fluid. You don't. You hang on for me in case nobody. What's the reason? Okay, well, let's say I'll give you a function for this thing. Okay, that's a protein that transports glucose. What happens if this is not fluid? Yeah.
Stuff to get in and outside of the cell, transport between
Right.
ions or molecules.
Right, so this basic, without the little aliens here, this lipid bilayer is quite impermeable to everything, except for small gases and really tiny things. So, do you need nucleotides to run a cell? Absolutely. And those get sucked in here, right? And glucose and lipids, everything has to go through here that you need to live. The thing that transports them are proteins, and they move. If this is an iceberg of lipids, they don't move. You die because you can't get glucose or any of the nutrients, or this cell can't respond to any of the signals like insulin that your body is producing. That's why it's fluid. To make these proteins have their function, this must be fluid. Now, can it be too fluid? What happens to you if I heat you up to 200 degrees? You burn, but what happens first? These will melt, and all the proteins will be released, and again you lose function. So we've got to have a fluidity buffer in there. It can't be too fluid, but we want to be fluid enough that we allow these proteins to function. That's the core message of the fluid mosaic model of the cell membrane. Okay, these are our old friends. Just understand that lipids are not just some boring, well, they are boring, but it's not just only that they're boring, but they are a functional part of that cell membrane. Okay, they actually have specific functions. They have different head groups. Most of them are attached to a glycerol backbone, but there's another backbone that also forms phospholipids. It's called sphingosine, and it actually takes the place of this hydrocarbon tail backbone, and doesn't have the second lipid. It actually has another one that's attached to it. So sphingosine is another polar head group, and actually has a tail part to it. So there's lots of different phospholipids, but the take-home deal is this part of that phospholipid is polar. So that's why it's facing to either the cytoplasmic or what's the other side called? Cytoplasmic side. So there's a cytoplasmic leaflet, and then there's your book strangely calls it a non-cytoplasmic leaflet, but there's a name for it, it's an exoplasmic leaflet, faces the outside. So you need to know why this, what the structure is and how it is used to construct this monstrosity. Okay. So this is a scale model of a lipid bilayer. Okay. And the key points to this thing is that the interior of this is completely disorganized. Those lipid tails are all bumping into each other, and that's what creates the fluidity, is those lipid tails cannot pack very tightly together. It also gives it the property of being hydrophobic. This is the permeability barrier right there. Okay. So here's the plan. The cell goes, I'm going to have to coat myself with this thing, and I'm going to make it completely impermeable, almost, not quite, except to the things that I really want. And then to get those things, I'm going to put proteins in through this membrane and take in only the things that are important to me. That's the plan. That's how it evolved. Okay. What's this stuff? This is the polar groups. The glycerol backbone, there's a phosphate, phosphoester bridge. That's charged, negatively charged. Okay. The glycerol backbone has no charge, but it's polar, so that's part of this red stuff. And then there's a head group, like choline. So when you see the word phosphatidylcholine, that word phosphatidyl refers to the glycerol backbone and 2 fatty acid tails. You should know that. And then the little head group is what you name it. So phosphatidylcholine means the head group is choline. It's one of these little guys at the top. Okay? The important thing is this is a functional surface. There are proteins that read the surface of that polar molecule. Okay? This is not without information. So there are a class of proteins that we'll talk about next time that are called peripheral proteins. They recognize this bumpy surface. It is quite capable of being read. Okay? And I don't mean read. I mean you're reading it, reading the surface. Okay? And then, of course, the water is out here. So we've got different molecules are going to partition based on where they fit into this picture. Right? That's real straightforward. Okay. Now, I think what we're going to do is, I think I skipped over, should have been some questions, okay, somewhere. Let's go back to the other PowerPoint. I guess we have about 8 minutes. Okay. So get your clickers out. There they are. Okay. Okay, so look at this one on your own first. Polling is closed. There's 5 answers there. Okay. Polling is open. Don't click yet. Okay, so try to answer. Let me expand this. Oh, get to do this. Okay. Move this out of the way. Try this on your own first. And then you can go ahead and click your answer, because I need an individual click, and then you're going to have a group click. Sneak this in. Use this timer thing, so let's see. Give ourselves 30 seconds. So I would go ahead and click something. You can don't click it. Okay. You guys like answer 3. Okay. And so let's look at answer 3. That's C. Okay. The bicoid messenger RNA is selectively carried by a cytoskeletal protein to the anterior pole. Okay. Let's look at these others. After translation, the bicoid protein is selectively trapped at the anterior pole. Doesn't that happen? What's wrong with that? Anybody? Yeah.
Isn't it just the one cell, so it doesn't trap it?
It actually does trap it. So actually the full answer is not on here, but everything else is incorrect except for this thing right here. Right? It actually is transported. But what's wrong with this? It's the mRNA. It says after translation, the protein is moved. That's not what happens. The messenger RNA is trapped. It's moved and trapped. Okay. And before translation, the bicoid diffuses randomly but is selectively trapped. That's not what happens. It's actively moved by along a microtubule. Okay. And then figure out the rest of these why they're bad. Okay. Now let's go. Let's do one more. Colon is closed. Okay. Put this up here. And again, let's do it by yourself. Colon open. There are 5, I think. Sorry. Guess what? God, not doing well with the technology today. Let's forget about that. I give up. All right. So this is you'll have this type of question on exam where you have a single key. And you can use the answers once, more than once, or not at all. So it's not a one-to-one matching. This is not matching. This is, you might use all of them. You might use just one of them repeatedly, but all of those possibilities are true. Okay, let's do under hot conditions of high intracellular. So this is why I told you that physiology. First, get into your mind what it is that you think is reasonable to happen. So you have high intracellular iron levels. Aconitase dissociates from this region in the ferritin messenger RNA. What's the answer? Right, E. Right. Okay. Phosphorylation of IF2 blocks this process. Translation. This structure is added to different exons in calcitonin and CGRP. Right? Poly A10. Right. Okay. So, all right. I think that's enough for today. I don't know what to do. I really don't. The only thing I can tell is, can you come to office hours or anything so we can check it? Because I don't understand why there's not